morning all. Welcome back to the lecture on the amino acids and proteins. Let us look into the applications of these protein molecules as intracellular matrix, intracellular network, that means coupling of cells. In some other areas of our biological cells. Let us see what is the applications of these proteins in our circulatory system. As you know, circulatory system, just I will go through in brief about the circulatory system. When you think of an organism, there are two types: unicellular and multicellular. Unicellular means only one cell, single cell is covered by the cell membrane and cell wall, of course, for its protection. So the entire surface area, entire outer area of the cell is in contact with its surroundings, so that it can exchange the material with its surroundings for its survival, growth and survival. So it will take what it wants, the nutrients, and give away the waste to its surroundings. A direct exchange is possible, is accessible to its environment for an exchange of materials. But when you think about the multicellular organisms like our body, example human body, it is a large number, normally another large number of cells it contains. As you know, the body is composed of 80 organs, nearly 80 organs, and the types of cells present in your body, 200 types. They are talking about the types, 200 types of cells. And how many cells our body is composed of? Trillions, 38 trillions of cells, not billions, trillions of cells, very big number. So how can we able to expose, it's possible to expose all the cells to its environment for access to its surroundings for effective exchange of materials is highly impossible. So whichever design you propose, whether nature or our idea, it is highly impossible to expose all the cell to its surroundings. Therefore, the nature has given a gift or a design. It is called a circulatory system. So what is a circulatory system? It is a flowing of liquids, a fluid is flowing in a circular fashion. A fluid is flowing in a circular fashion through all the tissues, through all the organs, in a circular fashion, so that every cell can exchange their materials with the flow of the fluid in our body. What is that organ, or what are the compartments which can support that flow of liquid? What is the liquid, the fluid that is flowing through? The fluid is blood. Blood, I am talking about the chemistry of blood. Blood is of course a colloid. So let us discuss the details of blood in the later stage. We will see that is the system supporting for the circulation of the fluid. So the system here, I will just talk about this. This is the point. It is the heart. I will tell you later what is the heart, because usually written heart from this point. These are, that is called a tubular, circular, in tubular shape. They are called blood vessels. That is tubes. All over the tubes are spreading all over the body. Starting from this point is called as point where the heart is located in our body. As you know, the heart, it contains 4 different chambers. So I don't want to go in details about the chambers. The upper and the lower. Upper parts are called as atrium, and lower chambers are called as ventricles. So this in the diagram, it is the left part of the heart, this is the right part of the heart. Because whenever you write a diagram or draw the diagram of any organ, we write always the mirror image of that. That is a mirror image of this. Right becomes left, left becomes right. So it looks like what is that? Right. It is a left. Right, but it is a left of the heart. It is right of the heart. The ventricle. It is a left ventricle. This is right ventricle. Atrium. Right atrium and left atrium. So from this point, blood what is that? The vessels. So vessels are what is that? Extending from here. So major vessel, here major vessels. These vessels are extending to all the parts of the body. So it is branching out like a tree, large number. The branch out in large number. So these two are vascular system. So it is called as, of course, artery. It is called as a vein. A major artery, a major vein. Here branching out into a large number of branches to pass what is that? To pass through all the organs, because this is a system, or vascular system should pass through all the organs, branch out in large number. Here branch out in large number to reach each and every organ. That is important. So branching is there. There are multiple branches, but branches are not shown here, just a brief branching like this. Now they are. And finally, in every organ, what is that? Every tissue, this is a microcirculation. Capillary. Here branches into finally what is that? Capillary. A main branch it is called arteries branching out, is veins branching out. And reach here. And the capillary system like this. It is one circulation race, always in circulation. It is circulation, cycle back to point. From the heart, it is flowing like this in that to all the tissues, all the organs, and where here it is passing like in capillary, very small capillaries, so the size of the capillary, the size of these articles, and again flow back through these capillaries and again into veins and bigger veins, major veins, and back to the point again. This is circulation. Keep on circulating. That is back again flowing like this. And talking about this, there is the branches here. Blood is always flowing from this artery, flowing through from the heart through the heart, that is artery, and back into heart by veins. This is the direction of the flow of the blood. Let us talk in details about this. What is the systemic capillary? In every organ, in every tissue, that is, of course, the size of the vessel decreases. Here, bigger size, nearly about 2 centimeter diameter. Here, about 2.2 centimeter diameter. Of course, a little bigger than vein. Major vein is little bigger than the major artery. This artery, as down the order, the size decreases as spreading on, decreases, and comes to arteries, small arteriole by this time, and again what is that, becomes smaller and smaller, small capillary like this. A capillary bed, there is numerous, millions of capillaries spread, there is only shown like this. So it is spread into millions of capillaries like this, bed of capillaries. Small capillary, the size of capillary will be 10 to the power of minus 3 millimeter. So initially what is that, from the point of commencement it was 2 centimeter. By the end of the tissue, what is that, what will be the size of the capillary? 10 to the power of minus 3 millimeter. 0.001 millimeter size capillary, fine capillaries bed. So it is a bed of capillaries. So these capillaries again continuation like this bed of capillaries from this point. It is capillary, arterial end and venous end. That is arterial end and venous end. Commencing from there, continuation here commencing from what is that, venous system is commencing. Capillary again what is that, branch into bigger and bigger bracket. This is a capillary bed. In every tissue, millions of capillaries divided so that it can have access to every area of the tissue. That is why we have a better capillary. It's called systemic capillary. It is known as a bed of microcirculation. It is also called microcirculation. So blood flowing from here and flowing through this, all these capillaries, and flowing back through veins back into heart. And look at that in the flow of blood now, the pressure. What is the pressure experience is 120 to 80 millimeter. And this 120 to 80 millimeter, that is 120 systolic pressure, 80 diastolic pressure. That is when the heart compresses, and it is maximum healthy person 120 millimeter of mercury, and minimum it is after relaxation 80 it becomes, that is pumping in the area in this area, and here also 120. While the blood begins to flow down, the pressure decreases. Comes here, arteriole near the beta capillary becomes 32 millimeter. While the blood is spreading or flowing through capillaries, here through venous side, here venous side further there reduced now 12 millimeter. Variation of blood pressure all along in the circulatory system, beginning 120, afterwards now it will go down to 80 after relaxation. This 120 reduces to 32, and in this area 12 millimeter pressure, and we are coming back now 5 millimeter, that is major vein. Look at the difference in pressure. Just talk about the pressure. 120 to 32, blood flow from higher pressure to low, from ear to ear, from this point of heart to every organ. This I am talking about only one organ. Every organ is nearby 32 millimeter. Blood flows, that is flows by itself because of difference in pressure. And while flowing back now, it is 12 millimeter, or flowing back 12 millimeter to 5 millimeter pressure. So the difference is less. Even then, the blood is flowing through because of it has the valves as well as the smooth muscles surrounding. Both will cooperate. In addition to the difference in pressure, very small, will cooperate the flow of blood back into blood. The smooth muscles will action, mechanical action will make the blood vessels to dilate. That is dilation and constriction will make a flow in this back into the heart. In this capillary system, I am not talking about the systemic capillary. What is that? That is now you think about this is what I am these. That is the organs I am talking about. That is organs in which circulation is taking place. What are these organs? The organs are just I will brief about the organs connection. This is of course this heart is connected with the lungs. So circulation is from this chamber to this chamber via lungs for exchange of materials, oxygen and carbon dioxide. Oxygen is a nutrient, carbon dioxide is a waste. So for exchange and exit from the lungs. So that is why for this purpose this circulation also there. In addition to this circulation like this connection to this circulation is also in connection with two more organs, that is GIT, that is small intestine, from where the blood blood collects the nutrients required for the cellular activities and nutrients for the cells from the GIT, and also it is connected to the kidney, very important. Just like a circulation is also kidney, so where it gives the waste product collected from different tissues. The waste products, of course, let us discuss later in this. These are the waste products. So this is what the circulation, because I have shown only the lungs connection with the heart, and other two organs, GIT and kidney, which is connected with is not shown in the diagram. Of course, understood circulation system. And this circulation is common in all the organs, it is in all tissues. In addition to this, to refresh the cell all the time, to have their nutrients and take out the waste. So now think about this system. There is a flow, there is a possibility to flow blood from this point to this point, like this, and back to this point. Now the important is, what are these? Just talk about this organ art. Talking. Just a brief account of this art. The cells of the heart, cardiac cells, which arises, which are actually differentiated from the embryo, is the stem cells during the embryonic development itself. These cells have been matured, the cardiac cells have been matured, have been specialized, have been differentiated from the stem cell of the embryon. So during the embryonic stage only, and form an organ like this. That is heart, cardiac pump. And these muscles are cardiac muscles are highly specialized for the function. What is the function? So the function of the heart is that there is always an equilibrium between the amount of energy generated by the heart for pumping, and the amount of energy of human body is demanding to pump the blood to all the organs to have the nutrients, sufficient nutrients for all the cells. So there is always an equilibrium between kind of equilibrium. In science, equilibrium, state of equilibrium is very important. So for this activity, there is also muscular, circulatory, for this circulation, action of circulation, there exists here always a kind of equilibrium. The equilibrium, whatever the amount of energy body demands for this complete circulation, the same amount of energy should be generated by the cardiac cells. That is a specialization. Sometimes it demands more, sometimes demands less. So when the body is exercising, usually demand is high. When the body is rest, demand is less. That is the kind is there. The adjustment is also there. So that is a kind of adjustment, equilibrium. The heart should be in that position. That is a kind of specialization of the heart muscle, that is heart cell. And that too, the specialization is that, but for this, what is that? It is a continuous process. Blood flow is always a continuous process. So for this, the continuous generation of what is that? These cardiac cells, continuously synthesize ATP. As you know that the muscle cell keeps synthesizing ATP for a muscular action, where the ATP, the chemical energy, is getting converted into mechanical force. Your mechanical force into a mechanical pressure. There is a continuous work, compression, expansion, compression, relaxation. Is a continuous process. Therefore, continuous synthesis of ATP molecule is important for the continuous generation of mechanical pressure on the blood for a flow. That is important. That is why. So these cells are specialized. Cardiac cells are specialized. What kind of specialization? They have a flexible metabolic activity, flexible metabolic mechanism. What is a flexible metabolic mechanism? It is the one, what is that? The cells are requiring, what is that? A metabolic fuel for the synthesis of ATP. What are the metabolic fuels? So this is the cell is very different from what is that? Other cells. So this is the one which can use any metabolic fuel available in the tissue. So it may be glucose, fatty acid, ketones, or what is that, lactate, or any carbon substrate. So it will not differentiate. Usually some cells ask only particular fuel for this ATP synthesis, but this is what is that, the cardiac cell is specialized to what is that, switch on to use any fuel available at that time, because there should not be any stoppage of synthesis of ATP, and it leads to stopping of heart and stopping of blood circulation and the end of life. That is why the cardiac cells are highly flexible in using metabolic fuel. So as you know that the glucose is available in plenty after postprandial state, but during fasting glucose may not be available, but fatty acids in place, fatty acids available, amino acids available, and if amino acid is not there, if fatty acid is not there, they can, what is that, depends on what is that, ketone, sometimes ketones available, sometimes lactate or some other any carbon substrate available to it, you take it, and the mechanism, what is that, mechanism of synthesis is different, so it switch also that switch on to that mechanism, make ATP. That is a kind of specialization of the cardiac cells. Right? That is why the heart is specialized, it is continuous, otherwise will not be suitable for this application. Continuous circulation. So that is a kind of specialization. That is only a brief account of the heart pump. So there is always a generation of what is that, some kind of mechanical pump. So the cellular activity, what is that? All cardiac cells have the mechanical function. What is that? All cardiac, usually muscle cell, no, the cardiac cells, a muscle cell. The action is first is compression. So cellular compression. This compression is all supported or play important role. That is the protein molecules of what is that, intracellular framework. So this is the intracellular framework actin when intracellular, what is that, matrix, which are made by proteins or what is that, just recall that the previous class, thin filament, intermediate filament and microtubules, they construct that intracellular matrix. So these are playing important role in the contraction. The filaments, intracellular matrix, intracellular framework plays an important muscular contraction. So that is, I don't want to go in detail about. This is a kind of specialization, continuous process. And think about this. What is that? About this organ, the vessel. What is blood vessel? This blood vessel is artery. This blood vessel is vein, and one more is there. Let us take about lymphatic vessel. A brief about this lymphatic vessel. Whether it is artery or what is that, vein or lymphatic vessel, all of them have what is that, structure. What is structure? What is that? Let us look into a brief structure of this vessel. That is blood vessel. So there are blood vessel, 3 layers. Blood vessel is made out of 3 layers, 3 cylindrical layers. Inner cylindrical layer and middle cylindrical layer and outer cylindrical layer. That is a layer, tissues. This is a tissue inner. Next, surrounded by another tissue, cylindrical tissue. It is surrounded by a layer of another thing. This layer is called as tunica interna. This layer is called as tunica media. This layer is called as tunica externa. They are called tunica. There is different names. Cylindrical layers forming a blood vessel. And just a brief account of it is what is important. What is this tunica interna? That is what is that written later. What is composed of? A basal membrane in which the cells are embedded. Cells are laid. These cells are important. These cells are called endothelium cells. They are called as vascular endothelium cells. Shape, look at the shape. Flat, squamous, epithelial cell. Endothelium, just like now, skin, epithelium, but they are very soft, not like a skin. Skin, they have different proteins in the cellular matrix. Here, protein molecules. The matrix is there, protein molecules are different. I will take the properties of this later. These are shaped, look at the flat, thin, squamous, very thin, flat like sector, very thin. Here, layer surrounding. Actually, this is like a sheet. That is a circular sheet. What is that? A tubular sheet. Forming a tube like this. Sheet containing a layer of endothelial cells like this. All sides surrounding. So luminal side, inner side. Annular side, what is that? What is that? A cells, all cells are in touch with cell. There is a tube is whatever going on, always in touch with cells. A layer of cells. A thin layer of cells. A sheet when you want it just into a tube like this. You form a tunica like this. Endothelial cells. I'll talk to this later. And the next layer is what is our basement layer. This is of course a thin layer, a tissue like this, tissue on which these cells are fixed. But this is exposed. These cells are exposed to material. What is that? The blood flowing. Bigger here, smaller here. And tunic on media, one more layer. That is a thick layer, of course. One more layer. This layer is a tissue made of smooth muscles and elastin. Elastin is very important. Elastin is a kind of protein molecules come out of intermediate. Intermediate, what is that? Filaments. Elastic, smooth muscles and elastin. And I'll talk to this about this. This is the content of smooth muscle and elastin varies down this from here to here. I'll tell you what the purpose of this. Next layer is tunica externa. This tissue is made of collagen and elastin as well as. Because elastin is important. This is also some of elastin. So elastin is present there. So elastin is because of what is that? Blood requires. The blood vessel undergoes dilation and constriction all over. While blood is flowing, dilation and constriction. For this dilation and constriction, that is flexibility is more important. This flexibility is all because of elastin present here. Elastin. Now in this, the thicker layer, it contains smooth muscles and elastin. In this area, wider area, here more elastin is present, less amount of smooth muscle tissue. Down the order, it is interchanging. Elastin amount decreases, muscle amount raise, muscle tissue increases. Down this. The reason here the area is area of high pressure. Here also area of high pressure. 120 when it is exerted, 120 here. This aorta artery is 120 here, the main artery all over here. So because of high pressure, this vascular tubes should withstand, because the pressure, the fluid exerting pressure against the vascular wall. So it should be able to withstand the high pressure. To withstand high pressure under what is that? Dilation. That is why you see that artery always keep every pump. Artery what is that? Dilate, expand, then compress. Dilation is high pressure, dilation. So this dilation, what is that? It requires more elastic material. More elastic material can withstand pressure. Which can, what is that? Because of dilation. The pressure what is applied now, it will go in form of dilation and withstand like this. That is why we have more elastic. Down the area, what is that? Down the harder, the amount of pressure decreases. There it doesn't require more elastic. It requires more muscle for further support of the flow of liquid. More muscular action, the mechanical action. No. So for the blood to flow, smooth flow, that is why we have variation of the content of what is the tissue mix, that is smooth muscle tissue and elastic, all along down the order is this. And up to this there are 3 layers are there. From then onwards, here in this microcirculation, this further, that is a branching out into a bed of what is a capillary. These capillary vessels is not having these 2 layers. It is only this inner layer, that is basal membrane with only cells. The reason here, this tube, inner tube, is very leaky, highly porous. But these layers will prevent what is that? Porosity, that is leaking of blood. So from here to here, there is no leakage of blood. Flowing, what is that? Blood is flowing from all to different organs. Till that, what is that? Start of the organ, no leakage because of these 2 layers. When enter into what is the tissue, it is in the form of only this microcirculation. What is that? This tube, what is that? First tunica interna, what is the different capillary system. So this is a leaky. The reason here requires what is that leaking of the blood fluid through all tissues. Here also leakage. So both these what is that capillary? What is that? These are capillary arterial capillaries and these are lineal capillaries. Both these capillaries are leaky. Here, when the blood is flowing through because of pressure, what is that? Fluid is come out. That is, fluid of the blood is come out and sweeping through the tissue. I will tell you what is the leakage, how the molecules, what molecules are come out of this leakage. Not all molecules. Only some materials come out, leak out from the blood capillaries and water as well. So this is a fluid, some fluid, right? Some fluid is come out and wash the tissue, entire tissue. Wash on the end of is a tissue matrix. So tissue matrix containing fluid and tissue matrix in which cells are embedded is a tissue matrix. Cells are embedded. Now we have the fluid. What is that? We can realize different compartments of fluid. 3 different compartments, fluid compartments. Here, fluid inside the cell, fluid outside the cell, that is the interstitial phase, intracellular matrix, extracellular matrix. Here, matrix outside the cell, then tissue matrix, that is tissue matrix, cell matrix, and also this fluid. What is that? This also liquid is there. So we have different what is that compartment, fluid compartment. So vascular compartment, tissue compartment, and cellular compartment. 3 different compartments of what is the fluid compartment. The fluid is flowing from this vascular compartment to tissue compartment, and from tissue compartment to cellular compartment where the exchange takes place. And again back, what is that? This fluid, that is this fluid now entering was the fluid is sweeping through, washing, refreshing all the cells, refreshing the entire tissue matrix, washing the entire tissue matrix, and what is that? Sweep away and flow into these capillaries, the veins, and back into veins. That is what the flow. So in this kind of flow, what is that? Movement of fluid. Here the blood, the fluid. So fluid along with the components needed for the tissue is what is that? Leaking, flow out, and what is that? Refresh and flood and wash, then enter into venous system and back into this. In this, that is the sweeping of this tissue. Look at that, the water, and I'll tell you what is that blood, also the contents of the blood, and contents of the blood, the nutrients. Nutrients are glucose, amino acids, fatty acids, minerals. Minerals are sodium, potassium, calcium, phosphate, chloride, that is, and carbonates, minerals. So, and also oxygen, nutrients. So these also come out, these are leaking out. And the blood contains, what is that? And then talking about the blood chemistry, blood is a colloid. Colloid means in liquid phase, these what is that? These materials are dispersed. The size of the particles dispersed was is more than 100 nanometer. The size of what is that solute particles, if dispersed one is more than 100 or 200, it is known as colloidal, 2-phase system, liquid phase and solid phase. And also some of them are liquid as also, that is given dispersed also. So that is a kind of what is that is called as colloid. That is a colloid because in our biological system, whether it is a liquid medium or fluid medium or a gel medium, they are all colloids. Gel is a colloid and blood is a colloid. So it's a colloid chemistry in our biology, very important. That is, blood is a colloid, but the solid disposed in liquid. Gel is liquid disposed in solid. There is only difference, colloid difference. So now we talk about this, the things which come out from this. What is that? Capillaries. I'll tell you how the leakage is possible here. So these are all the materials. Oxygen, amount of this. Plus the blood, no, blood contains RBC, WBC, platelets. These are cellular materials. RBC cells, WBC cells, all cells of what is that? WBC, white blood cells, RBC cells and platelets. That these different cells are there. This RBC are to carry oxygen from the lungs of the blood into the tissues. RBC, the purpose of the RBC cells, what is that? Carry oxygen from the lungs into different tissues. And the purpose of WBC, what is that? Blood cells, to look into that any infection over there. I'll tell you what is mistake. And platelets, these are the cells or cell debris. There is cell, fragments of cells or sometimes we have like a cell. These cells have no nucleus. These cells have nucleus. These cells have no nucleus. These are three different cells are there in blood. And these platelets, what is the purpose of this cell? If any damage should be what is that? Plugged immediately. The plug formation, because this is the platelets will change. These platelets cell into clot by releasing, that is, fibrous proteins and subsequent cross-linking make a clot to plug the hole. That is the purpose of the platelets. So it requires plugging, otherwise blood leaks out into tissues. That is the purpose of the blood. And WBC, you know that this is for immune system to attack and remove, nullify the effect of all infectious microorganisms present in the tissue matrix. So these cells are present and protein molecules are there. These protein molecules are globular molecules, albumins and globulins. Albumins and globulins, big molecules, globular molecules, and the purpose of this to maintain the osmotic pressure of the blood. Because this also, albumin, globulin also are a carrier of some materials which are not soluble in water. To carry that, can combine with them and carry that materials. So the purpose of this protein, what is the blood protein molecules, globular proteins, they are present in to make up the osmotic pressure, to maintain the osmotic pressure of colloid. Colloid means should have osmotic pressure to balance the water in the compartments, to balance the water in the vascular system, to balance the water in the tissue compartment, balance the water in the cellular compartment. And also blood contains vitamins. Of course, these vitamins, what is that? Requires to enter into cells. So these are the vitamins of the different vitamins are A, D, vitamin A, vitamin D, vitamin E, vitamin K, vitamin B, vitamin C. These are water soluble, these are fat soluble. But these vitamin molecules are important in the metabolic, what is that, mechanism of some metabolic reactions. They are not directly involved, they are in coordination with the enzymes as a coenzyme of some important biological reactions. Very important biological reactions in which, in addition to enzyme, you require a coenzyme. These molecules, that is vitamin molecules, act as a coenzyme in that mechanism, without which the reaction is somehow not efficient, not produce 100% products, what you realize. So that is why we need all these vitamins. That is, of course, as a source through food material. These are water soluble, these are fat soluble. So these are fat soluble, actually not soluble. What is actually water now? So water is a solvent, but they are not soluble. How they carry in this? Because these vitamins combine with albumins, globulins like this. They combine from a complex. The complex is water because these are water soluble. These protein molecules, no, globular water is highly water soluble. Even the big molecules, macromolecule, a globular molecule, water soluble. They can combine with for a complex is soluble because combine with this, they can carry the purpose of this. So I have real purpose as atmospheric pressure and carrier. The proteins are carrier like this. So BC directly soluble can carry with the water. In the soluble state, they will go along with the blood. And other components are hormones. Hormones are also present. These hormones are not from the nutrients. These hormones from the body itself. These molecules are synthesized at one site, but actions are different at other site. See, in one area they are synthesizing in some organs, that is glands, endocrine glands, about 12 endocrine, 7 to 12 endocrine glands are there in which these hormones are synthesized. But action is not in that organ. This action is in different organs. That is important. That is what is that? A gift is like that. Hormones. So these hormone molecules, after synthesis, they enter into blood. They travel in blood. So here also we have hormones. There are two different types of hormones: fat soluble hormones, water soluble hormones. Some endocrine glands release hormones which are only fat soluble. Some, that is hormones released by water soluble. So water soluble means they are soluble in the water solvent and carry along with the blood, carrying the blood. But what about the fat soluble? They are also called steroid hormones. That is steroid molecule. A steroid molecule, it is not water soluble. It is only fat with steroid hormones. Some estrogens like this, no. So steroid hormones which are not fat soluble or oil soluble. So these are the one carried by albumin, is combined with albumin and form a complex. The complex overall is water soluble and carrying the carrier. Therefore, these two are, in addition to maintaining osmotic pressure, carrying along with what which cannot carry the blood. That is fat soluble hormones, fat soluble vitamins. That is the purpose of these hormones. So this is the composition of what is the blood. So these things are also there, and these things are also present here. These are metabolic waste. These are metabolic waste released from the cells. So cells can exchange. That is, fluid is there, fluid compartment. Between these two compartments, cells can exchange. So here the blood give all this, except this. These two and these cannot pass through. Porous only for these. Porous only for these. But this porous is not for these molecules. These molecules remain, pass through without leaking. Pass through without leaking. All these cells, all these protein molecules never come out of the vascular system, remain in the vascular system. It is leaking only for these things, leaking for these, leaking for these, leaking for. Two gases are there, most important. This is most important for what is that oxygen, for the survival of cells, synthesis of ATP. So all cells survive on the synthesis of ATP. It is all because of oxygen, which oxidize the metabolic fuel to release ATP. So with the releasing of ATP, the byproduct, what is that? Is carbon dioxide as a product, waste product. So this is a nutrient as a reactant, and the product carbon dioxide come out along with ATP available for what is that mechanical, what is all biological reactions ATP is using. Any kind of biological expense. So biological reactions at the expense of ATP, or any mechanical reactions at the expense of ATP, all synthesized because of oxidation of what is that metabolic fuel. It may be glucose, fatty acid, anything like that into carbon dioxide, waste product coming out. So along with this waste also coming up, this is a metabolic activity, the metabolism of protein molecules. Sometimes protein molecules use a metabolism. Ammonia, urea, uric acid, creatinine also released as a waste. These are the waste, metabolic waste come out of cell into the tissue. And these waste will be taken away by this. veins. So we will give this, take away this, and water also wash. Water is also flowing, come out and flow. So this is what leaking only water and these things. These things from the tissue enter into these capillaries, and these things what is that leaking out. These all these things leaking out. See these hormones what is that I told you. These hormones are important. Some biological functions. That is the regulatory bodies. That is maintaining what is that some constant biological functions. Example, constant composition of all these blood compositions and constant pressure. Maintain constant is 128 like this constant pressure, and any variation again what is that? Sometimes it requires variation. So in that also the hormones plays regulatory bodies, hormones. So hormones what is that? Any variation of the glucose, that is hormone is what is that? Insulin releasing to maintain what is that? Glucose concentration increase means to maintain the concentration. Hormones very important to balance. There is always a constant composition, constant concentration of all these things in the blood. To maintain this constant concentration requires regulation. Any fall, these hormones are released to make it up. Go to the site, raising at one side, go to the site and do the action. That is called hormones. That is the purpose of the hormones. So different stages, but growth and development also required. At different stages, yes, sir, hormones are what is that? Organs are developed because of the hormone release. That is hormonal functions. So blood also contains hormones. Whenever in need of hormones, blood contains more number of hormones, more concentration of particular hormones. So these are the constant composition of the blood. And this is one more I want to talk about. While washing, while the tissue is washing, that is sweeping, when the fluid is sweeping through, imagine that the fluid come out and fluid pass into capillary system. But we see that it is not total in total. Whatever fluid come out, the same fluid, same amount will not flow. So about 85% of fluid will flow into venous. Remaining 15% will left over in the tissue micro circulation. Remaining 50% will circulation unavoidable. What is that? While the fluid is flowing from here to the circulatory system, the systemic circulation in all tissues, the fluid while washing away, only 85% of it will be taken away. Remaining 15% will remain. That 15% remaining in the tissue, what is that? It will remain over there for every circulation. Then is a problem for the tissue. Tissue swells. It is also called in medical terms edema. That is of course. That is tissue swelling and go for what is that? Malfunction in the tissue. That is why we need to remove that remaining 50% at the same time. There is a different circulation system. That is what I call this lymphatic system, a lymphatic vessel. The vessels or tubes are just like these arteries and veins. But it is not circulation, it is one-way flow. Blood is circular flow. What is that? Commencing from the tissue bed. Commencing from the tissue bed. Just like this. Capillaries. There is millions of capillaries. They are called as lymphatic capillaries. Closed vessels. Again branching into this. The purpose of this capillary to collect, what is the collect? Collect remaining 15% of the fluid from the cellular, what is that? Tissue bed. That is what is that? Modified like this. There is a specialized like this. Remaining 15%, what is that? Will collect it from here. That 15%, while collecting 15% of the water and some ions, along with collection, these vessels also collect, very important, in the tissue matrix may contains some cellular debris, some dead cells, or what is that, some cancerous cells, or microbes, presence of any microbes, virus, bacteria, fungus, maybe contains tissue, may contain sometimes, it is not contaminated. So that will collect now all those microbes, and also that all the white blood cells. What is that? WBC. RBC platelets will remain in this, but WBC, what is that come out of this? Leaked out like this. WBC, that is all white blood cells are come out. I will tell you what is that, how they leak out. WBC, what is that? Through tissue, that is, see that any infection are there, they'll come out. And in addition to collection of all this waste material, 15% of fluid left over, and waste debris and dead cells, there are some microbes, they collect over WBC. All what is about 5 to 6 that is cells are there, white blood cells, all of them are collected into this, migrate, what is that, diffuses into Q, and what is that, flow. The flow is called as lymph. Lymph contains all those waste, and flowing through, what is that, there is one more circulatory system. This lymph is flowing through lymph organs. What are those lymph organs? The lymph organs are lymph organs, there are 3 different lymph organs are there. Lymph nodes. Lymph nodes. So lymph nodes, about 800 lymph nodes. Lymph nodes are bean size in cluster. They are distributed all over the body from the neck to groin. Lymph nodes. So those lymph nodes are also there, and also spleen, thymus. That is lymphatic organs. Spleen, that is just below the stomach. Spleen is there, and in the chest, what is that? The thymus is there. There are lymphatic organs through which they are flowing. Where the blood cells, what is that? WBC are also there. They clean up. What is that? They what is that? Break any what is that? Any cellular debris or any dead cells or cancerous cells or any microbes. They kill, and what is that? Break into pieces and remove as waste, and back into what is that? Again, what is drain into. Finally, what is that? Flowing through all these lymphatic vessels. That is, a lymphatic circulation is there, flowing through all the lymphatic organs, and drains into finally what is that? The major veins near the heart. Back. So they fluid 15% again back into the, because the volume should be maintained constant. For a healthy person, any imbalance is that is because of some pathological conditions. So that is why you always see that in this circulation, in this microcirculation, 85% will be collected by the veins. Remaining 15% of fluid is collected by the lymphatic vessel along with any dead cells, any cellular debris, any cancerous cells, any microbes, and all white bloods which leak out from this are collected in this and back into this. That is why. So this circulation is important. So this is a special circulation, very important. We need a failure in this lymph circulation, the failure of also body. This is weak circulation as this lymph circulation. So this is our circulation I'm talking about. Now coming back, what is the importance of the protein molecules? I want to talk about this leakage. First I will talk about leakage. How the leakage taking place in this porous? What are the porous? It is a real barrier. Here in the what is that? In tube, all covering with cells. There is squamous epithelial cells and smooth cells, smooth surface. So this is what is that? A flexible muscle. What is that? Cells. Very smooth surface area. What is this? Gives a smooth surface for a smooth flow of blood without any friction. That is cells functions. And the porous, which is porous, this is the junction between them is a porous. Between cell, this is porous. Tissue is always a porous, highly porous. Tissue material is always, because thin tissue is there, is highly porous. This junction, here sometimes in different tissues different gaps are there. More gap, less gap. This junction, and also the junction I'll talk about later. This also the pores through what is that? The pores in the cell itself. These pores are because of this protein, what is the channels? Protein, the channels made out of protein molecules. That is a cell like this. So channels are also there. And leakage through this, out as well as in. Water soluble. First talk about water soluble means these are the water soluble materials and fat soluble materials. Here, water soluble materials are what are water soluble materials? These are things, all these things water soluble. This is not water soluble. Here, water soluble vitamins, water soluble hormones. They are all leaking through this between the cell as well as the they are called as fenestrations. Here, exchange between the gap between the cells. Water, here water and water soluble. So between here water soluble and fenestrations. Here fenestrations are there. In some organs we have fenestrations. These what is the channels made from the protein molecules built inside this channel. Because water molecules cannot pass, because this is always cell. Every cell is a lipid bilayer, oily. This lipid bilayer surrounding this. A lipid bilayer like this. Lipid molecules here. Surface is water soluble. This surface what is outer surface. Outer surface water soluble, inner surface water soluble, because this the middle layer is oily. So inside is oily or lipid fat soluble. So through this water soluble cannot pass through directly. That is why we need special channels or fenestrations for the flow of water soluble and water. So that is the way water soluble can flow like this. Waste as well as nutrients. And one more, water insoluble, that is fat soluble, directly what is that they can migrate or what is that diffuses through the cell. This and fat means like this, fat soluble and fat soluble, because fat, the atmosphere is fat, the layer is fat, fat can pass through fat soluble. So directly what is that, it is diffuses directly through this fat soluble. Oxygen is what is that, water insoluble. What is carbon dioxide? Water insoluble. All of them are water soluble. The waste material now is going in. Carbon dioxide and water. The carbon dioxide, oxygen are water insoluble. They are not water soluble. So they can what is that, fat soluble. They can what is that, diffuses directly to cell membrane. So diffuses like this. From here to true cell is the diffusion. So diffusion is quick because of the water soluble. There is fat soluble. Carbon dioxide, what is that, make an entry into oxygen, what is that, leaving the gas compartment. That is saturation. So that is one way, other way. And one more, sometimes what is that, the blood, from the blood we need what is that, from protein molecules pass out, important protein molecules like this, protein molecules like antibodies or some other protein molecules we need in this intracellular matrix. The protein molecules little bigger. Little bigger means small, little bigger, not bigger, medium sized protein molecules. In that case, they cannot pass through like this. They have a kind of this transcellular mechanism, forming vesicle like this, a bag of materials, protein molecules into a bag like this, again diffuse on this side. Here forming the cell now, entering from the inner to out. Here forming a bag of molecules, vesicle, fuse here and passes out. That is come out like this. This is called transcellular. In which antibodies, sometimes we need the antibodies for these that pass out or pushes out. So this is the way also is possible. So this is a different what is that, migration, different way of passing of the water on different contents, and also white blood cells, WBC cells now. These white blood cells also what is that, come out. They come out between the cellular gap. They are called sinusoidal cleft. So this gap between the cell is called sinusoidal cleft. So through which white blood cells what is that, migrate. Well, migration what is that, requires what is that, bending. We need bending of these cells and also bending, folding sometimes or twisting like this. Come out like this. It's usually bigger. These cells are bigger. These are RBC cells, are smaller. These are protein molecules like globulin and albumins. These protein molecules. These are RBC. RBC without nucleus. but WBC with nucleus. They are drained out from this. And only RBC and platelets and these albumin globulins will not come out of this. They will go through it. So this is what this we are talking about, very important. Concentrate here. Here requires what is that? Deformation. While the cells, when the cell is passing out from this vascular system into tissue matrix, requires some deformation. What is deformation? Cellular shape change. This cell undergo a little folding, twisting and bending, and these also little twisting and bending. They also undergo and pass out like this twisting, bending and twisting, bending. All this what is that very important. I talk about the importance of protein molecules. These protein molecules, what is that? Act as a intracellular matrix. So intracellular matrix, that is a flexibility, that is intracellular matrix play an important role for this migration of the cell with undergo deformation. So any kind of deformation in the cell, activity of cell is all because of intracellular matrix, as well as what is an intermediate filament, mainly inducing the flexibility in the cell. Therefore, if the cells are flexible, they can bend without any break. They can twist. Here requires some bending and twisting of this. Not only that, these RBCs, very important, while flowing through capillaries, they will not come out. This capillary size I showed you very narrow, that is nearly equal to size of this cell. While passing through, the RBC cells require what is that? Undergo deformation. Cellular what is that? These are the concave disc, like their shape is like concave disc. They are what is that? Undergo what is that? Some kind of bending, twisting and folding. While passing through a narrow, all this RBC, what is that? Undergo that kind of deformation, twisting and bending, because it had to squeeze. What is that? Squeeze through. The all cells are squeezed through because of the because they should have flexibility. So this flexibility will retain because it has a intermediate. What is that? The intermediate filament, that is protein molecules. That is the one induces flexibility for these molecules, these cells and these cells. So that is a requirement. Intracellular matrix plays an important role in this kind of movement of what is that cellular activity with undergoing deformation without any breaking. That is why I am talking about the importance of these protein molecules in these cellular activities. So this is what I am talking about, these cellular activities. And in addition to this, I want to talk about one more thing. This is about this. And here also, in the lymphatic vessel, here the lymphatic vessel, while the blood is what is that? While the contents are flowing into lymphatic vessels, one way is closed. How this lymph is entered into this capillaries? This is the way it is not is a capillary of lymph. It is also composed of the same kind of what is that epithelial. What is that? Endothelial cells. Same endothelial cells here. Difference here. This is a kind of arrangement in veins and arteries, but this is a kind of arrangement in lymphatic vessels. Look here, overlapping. The cell ends are overlapping. Look here, overlapping, and in between small gap is there. Flexible. These cells, what is that? Overlapping with what is that? Flexible gap can open and go close, open and go. So this flexibility is all because of intracellular matrix protein molecules. The activity of those intracellular protein molecules very important. So these when the lymphatic or lymph, what is that? Make an entry. We apply pressure on this, whether this cell WBC or what is that? Water, anything. What is any compositions or cells, dead cells? What is that? They apply pressure. When pressure is applied over here, when the pressure is applied over, no opening. When the pressure built up because of accumulation of what is that? Lymph in the tissue, this opens with little gap and enter. All these what is that? Lymphatic compositions, all the what is wanted now will enter like this and close. So flexibility, close the body one way. But once enter will not come out. Open for the entry and close. So this kind of flexibility is there. So this is the cells, what is the endothelial cells, and this kind of flexibility is very important for this, what is that, one-way flow, flowing only in that channel. As the lymph or fluid builds up, pressure and also muscular action, the constriction and dilation will make a flow of lymph. It is not because of the arch pressure, it is all because of building of volume in the lymph volume, and muscular tissue surrounding it action will pump these lymph back into the circulatory system. So this is the kind of mechanism. And what is that? We are talking about the importance of protein molecules staying here in the cell matrix. So this is one, and one more very important. These cells are derived. Very important. These cells are the one important. These cells can make. These cells can build. These cells can rebuild, repair these vascular tubes. How these vascular tubes are raised? How these vascular tubes have come or what is developed? It is all because of endothelial cells. Where do they come from? They have come from what is that? The stem cells at the embryonic state. Like that, no, or cell, other many cells are not like. These endothelial cells are differentiated or specialized, what is that, mature to form endothelial cells from the stem cells at the embryonic state only. Then, with the development of what, these vessels are also developed like this. How these endothelial cells are specialized to undergo what is a proliferation by itself. They multiply in numbers, very large number at a time. That is a kind of mitosis. That is a specialization. They can multiply or proliferate in large numbers. When they multiply and produce, what is that? They form what is that? Make a tissue, form a sheet. During proliferation, when the cells are produced, the cell will not breathe. They make a tissue of its own and form a sheet like this, a thin sheet like this, and form a vessel. This is the result. Tunica interna is what is that? A new vessel is forming. And once this this tube is forming, this initiate the formation of this layer and further initiate formation of this layer. So once it is initiated, or this layer or tube is arises because of synthesis, or what is the synthesis of these cells or proliferation of these cells and into this tissue matrix, this automatically built up with signals, corresponding signals. So this is how what is that? Blood vessels. All these blood vessels now. These blood vessels, capillaries and veins vessels and lymphatic vessels, all because of these endothelial cells. Endothelial cells have the capacity to proliferate and make new blood vessels, and whenever the blood vessel is damaged, usually it is by accidental when it is cut or damaged here. So damage first is plugged by platelets. Plugged by barring clot, and the clot afterwards, no, this gap is repaired. How it is repaired? Nearby endothelial cell undergo proliferation. Immediately, nearby endothelial cells undergo proliferation, multiply in number. With multiplication, what is the tissue? That is the sheet, they grow. This tube grow and fill the gap, repair the gap. This is how the migration. What is that? These cells proliferate and migrate to damaged area and make a new tube like this and repair and join it. Then what the plug will remain will dissolve back into the fluid. After the gap is repaired, and all what is that cell new blood vessel formation is all because of this endothelial cell proliferation and migration. This proliferation and migration is mainly supported by intracellular matrix of proteins. Migration, any migration of cell, any kind of immediate what is that synthesis, what is the proliferation of cell, it requires support of protein molecules which make the intracellular matrix. That is why I am talking about. Not only this, wherever the tissue is having hypoxia, that is concentration of oxygen is very low, not able to survive, where the nearby endothelial cells undergo proliferation, multiply in large number and migrate into the tissue and make a new cell, that is new vessel, to supply oxygen. That is also there. So migrate to any tissue from your proliferation and migration. Proliferation and migration all requires support of these protein molecules which are responsible for intracellular matrix. So that is why I am talking about the importance of these protein molecules here. The flexibility, migration and cellular deformation, cellular migration in the blood vessels, and blood chemistry. What is that? These are the importance of these protein molecules. Importance of the protein molecules in the intracellular matrix in the vascular system. This is a brief, that is very, I have been going very deep. Just a briefing about the importance of protein molecules. And thank you very much for this session.